Good afternoon, everyone. It's noon, so we'll go ahead and get started. Today, I'd like to introduce Tracy Zarrillo, who joined the station staff in March of 1992 as an agricultural research assistant 2, and has been steadily promoted up to technician 2 by January of 2014, working for Dr. Kimberly Stoner, where she assisted in the field and laboratory for research projects dealing with bee pollination, pesticides, floral preference trials, and wild bee diversity here in Connecticut. In addition to her bee work, she has assisted Dr. Chris Meyer and Dr. Claire Rutledge in natural history surveys of longhorn beetles, exotic insect surveys, and surveys of biocontrol agents for emerald ash borer throughout Connecticut. So she's got quite a bit of entomological experience here over her years working here at the experiment station, and traveled a lot around the state. She obtained her bachelor's at Southern Connecticut State University in 1992 and her master's at Southern as well in 2016. She was then promoted to Assistant Agricultural Scientist 1 this year, and she has published peer-reviewed articles in journals. Now, Tracy, as you'll find out today, is an expert on native bee taxonomy, native bee monitoring, and survey techniques and pollinator conservation. She's actually recognized as a regional bee expert by leading bee taxonomists that lead the bee monitoring efforts across North America, and has conducted bee identification workshops. I mean, she really knows her bees. So, among other projects, she conducts surveys for rare or threatened wild bee species in Connecticut, and is currently writing up a checklist of the wild bees of Connecticut, which I think is pretty close to almost done, right, Tracy?
It's getting there.
Getting there. Okay. So our talk today is a checklist of the bees of Connecticut then and now. Tracy, it's yours.
Hi, everybody. This is great. This is such a good turnout. Thank you all for coming today. It means a lot to me. Thank you. So back in 2010, I became interested in wild bee diversity in the state, and I started a wild bee monitoring program here. So this, along with other recent studies on Connecticut farms and forests, meadows, coastal areas, dunes, marshes, baseball fields, and sand barrens, has given us additional information about the bee species that live in Connecticut, the habitats that they live in, and plant-pollinator interactions. The culmination of this research is resulted in my upcoming monograph, an annotated checklist of the bees of Connecticut. So the topics that I'm going to cover today are: why should we care about conserving pollinators? What is a checklist and why are they important? The history of early bee research in Connecticut, an overview of bees in general. How did I come up with the checklist? What methods did I use? I'll talk about the results of the checklist that I have so far, and bees of conservation concern in Connecticut. So our environment has been manipulated by humans throughout history, with major events like the development of agriculture, European colonization, and the Industrial Revolution. However, the rapid changes of the past decades threaten the diversity of insects and other wildlife. I'm sorry, I'm nervous, so I'm stammering a little bit. This diagram is a global view of the impact of human-caused changes in our environment on insects. Some are direct and some are indirect consequences of our actions, like increased fire, storm intensity, and droughts as a result of climate change. Certain species of bees are declining in the Northeast, and some factors implicated in their decline are habitat fragmentation, pathogens, non-native plants, herbicides and insecticides, overgrazing by white-tailed deer, shrub encroachment due to fire suppression, loss of open forests and forest clearings, and loss of rare habitats. So why should we care about conserving pollinator biodiversity? 87% of all plants are pollinated by animals, such as bees, flies, beetles, etc. So the loss of diversity of pollinators would result in a loss of plant biodiversity and would have a critical impact on ecosystem function. In addition to pollinator services, pollinators are part of food chains and support many types of wildlife. One of the world's most useful tools for conservation is the IUCN Red List. IUCN stands for the International Union for Conservation of Nature. Their Red List of threatened species has evolved to become the world's most comprehensive information source on the global extinction risk status of animal, fungus, and plant species. The IUCN measures the pressures acting on species, which guides and informs conservation actions to help prevent extinctions. The vast majority of animals are invertebrates, and it's estimated that vertebrates only comprise 3 to 5% of all animals. Approximately 1.5 million described invertebrate species are accepted by the IUCN. However, only 1.6% of them have been evaluated, and of that, 27% are lacking sufficient information. In the graph on the left, you can see that the number of described species of insects towers over the number of described species of birds and mammals. And the graph on the right, you can see that the proportion of species assessed for the Red List is heavily skewed towards vertebrates. So in 2007, the US National Academy of Sciences reported that for most pollinator species, the paucity of long-term data and incomplete knowledge of even basic taxonomy and ecology make assessment of status exceedingly difficult. So we have a problem. How can we conserve when we don't know what we have? And that's where checklists come into play. So checklists capture a snapshot of the known biological diversity of a taxon that occurs or has occurred in a specific location at a specific point in time. The data from a checklist can be used to figure out geographic distribution of a species. Checklists can be used to help identify species of conservation concern. They provide a baseline for detection of change over time, and they can support modeling efforts for anticipating responses to climate change. These lists are useful for government agencies and universities, and they're also now being incorporated into citizen science portals like iNaturalist to help narrow down the identification of species for certain geographic areas. Connecticut is one of the few states in the US that has extensive historical knowledge of its bee species composition. Henry Viereck was the man responsible for writing the first checklist of bees back in 1916. Viereck was an avid explorer and insect collector back in the early 1900s and has been referred to as the Huckleberry Finn of natural history. In 1903, Viereck was hired by W.E. Britton, the first state entomologist here at the Ag Station, to help to organize the Ag Station's insect collection. Viereck also collaborated with Britton on a study of insects visiting flowers of orchard trees and fruits in 1904 and 1905. Drawing heavily on the insect collection here, in 1916, he published his monograph called The Guide to the Insects of Connecticut, Part 3, the Hymenoptera or Wasp-like Insects of Connecticut. So in his monograph, he presented Connecticut's first checklist of 172 bee species, 62 probable species based on known range and habitat, and he also included a taxonomic key. Importantly, he included state distribution, floral records, and phenology which allows us to compare not only what species have persisted in the state, but any changes in seasonality or distribution. The floral records also help us to see what plants were in the landscape at that time and what bees were using them. And then in 1920, W.E. Britton wrote an updated checklist for Connecticut, which included an updated list of bee species. The taxonomy of certain bee species was revised with new information since 1916, and that's one reason why Britton's total is less than Viereck's. Lumping and splitting of species is very common in the world of taxonomy. In 1938, Britton reported 31 additional bee species for the state. These works are of tremendous value. However, it is the specimens in museums that really tell the story. Conservation work really depends on the physical specimens behind the literature, because the understanding of some of the names in the reports has changed as we increase our knowledge. So before I go any further, I'm going to give a brief background about bees in general. Silas Boser of Washington State University created this wonderful diagram to show the lineages of bee families. Bees are a highly diverse group of insects and can be found on every continent except Antarctica. There are 7 bee families in the world and about 20,000 described species worldwide. There are about 4,000 named species in the US and Canada, and there are even so many more bees, particularly out west, ones that haven't even been described yet. Bees are closely related to apoid wasps, but they've evolved during the Cretaceous period to become their vegetarian cousins. It is believed that bees help to drive the evolution, diversification, and radiation of the flowering plants on which they feed. Bees use pollen, nectar, and sometimes plant oils for sustenance and larval development. Something else that's interesting is bees show a rare bimodal latitudinal gradient with the highest species richness at mid-latitudes. Xeric and temperate zones host higher species richness than tropical areas. There's large hotspots of species richness in the southwest US, Australia, and the Mediterranean basin into the Middle East. Contrasting with species-rich arid temperate areas, the humid tropics and even arid tropical areas are generally more poor in species richness. This strongly contrasts with other pollinator groups in many other taxa, who typically achieve their greatest richness near the equatorial tropics. Low levels of precipitation during the driest months and reduced seasonality and enhance high richness. And humidity may play a key role in limiting bee distribution in the tropics through spoilage of pollen resources for solitary bees. So before I talk about how our native bees live, I want to mention the honeybee, because that's the bee species that most people are familiar with. They live very differently than our native bees. They evolved in Asia and were brought here to the US by European settlers. They live in giant colonies and they overwinter as a colony, with the workers metabolizing the stored honey to keep the colony warm over the winter. Beekeepers have to make sure that there's enough honey stored for the workers to burn through the winter, and often supplement with sugar in the early spring. Honeybees reproduce as a colony by swarming and have intricate methods of communication for informing other members of food sources. Bees have a variety of social structures. Honeybees and some native bees are eusocial. Bumblebees are considered primitively eusocial because they go through an annual life cycle where the queens overwinter and are solitary in the early spring, and then form larger colonies with a division of labor over the growing season. Semisocial bees form smaller colonies and the division of labor is among females of the same generation. Finally, some species are facultatively social, meaning some individuals live on their own and some live in colonies. The next level is the simplest form of social organization called subsocial. Subsocial bees construct their own nest, but continue to provide care and protection for their offspring. In the photo on the top right, you can see a male bee in the genus Ceratina performing nest guarding while the female provides provisions for the offspring. I don't know if I can see. So that's the male there, and that's the female. However, most bee species live a solitary lifestyle and do not live in colonies like honeybees. The female solitary bee constructs her own nest and leaves a provision of food for her offspring. She does not get help from any other bees, and she usually dies or leaves the nest before her young mature. And then there are the parasitic bee species. These species have figured out that it's easier to invade the nests of other bees rather than collect pollen for themselves or for their brood. As such, these lineages have lost their pollen collecting hairs and sometimes look more like wasps. There are two types. A kleptoparasite will enter the nest of another species and lay an egg inside of a brood cell. The parasitic larva consume the provisions of the host species and most of the time will kill the host egg or the host larva. And then there are social parasites that enter the nest of a social species. They usually kill or disable the queen and then produce their own eggs, and the workers of the host species will tend them to maturation. So along with differences in social structure, bees also prefer different nesting areas types. Many solitary species nest underground, and the soil texture preference varies with each species. Some like loose sandy soils, while others like hard-packed clay. Some like bare ground, and some will nest under leaves, stones, or wood. Most construct a main tunnel with side branches which contain lateral cells, but others only have a single tunnel with a cell at the bottom. The females excavate the soil from the burrow as they dig, creating a small mound at the surface called a turret. These nests look much like an anthill, but the opening will be larger. And other species are cavity nesters. These will use pre-existing holes, such as beetle galleries in old wood, hollow trees, nail holes, etc. Others create their own cavities by excavating out the pith of dead branches and twigs. These types of nests consist of a series of linear cells with transverse partitions terminated by a plug at the entrance. Social bees like bumblebees will nest in places like compost piles, under rocks, leaves, or in clumps of grass. And then, while we're talking about cavity nesting bees, there are some cavity nesting species that use flower petals to form larval cells. And even though this species called Osmia ovaceta does not occur in Connecticut, I wanted to include these photos because I just think that these nests are beautiful. Now that we have some background about native bee biology and early bee research in Connecticut, I'm going to dive into the current checklist and tell how it all came together. So I started my investigation with records that we already have here at the Ag Station, at UConn, at Yale. I also checked online global biodiversity portals such as Bison, GBIF, and iNaturalist for additional records. I queried members of scientific societies in Connecticut to see if anybody had private collections that they wanted to share. And I used that information to see where we have knowledge gaps and made a list of undersurveyed habitats species that have not been seen in 10 years or more, species that should be here based on known range and occurrence in neighboring states, but have not been detected yet, and species that are hard to detect based on life history. Our bees here at the Ag Station have been digitized into the arthropod easy capture database, which is housed at the American Museum of Natural History in New York. We're soon going to be moving our bee data into another platform called SCAN, which will allow all of our records to go live on global platforms like GBIF. To give you some idea of the numbers of Connecticut bee records, here are some totals. These are totals from the arthropod easy capture database and do not include private collections. The first total includes all institutional collections from the various museums around the country. The next total is the number of historical CAES records, meaning records that were already here before we began our bee work in 2010. The last total is the number of bees collected in our contemporary projects, including bees from our lab and Chris Meyer's lab. We've added over 16,000 new specimen records, which are now being recognized by other researchers in the US and Canada as a valuable resource for taxonomic studies and revisions. There's been a big push to revise certain bee genera in North America that have caused taxonomic confusion historically. And knowing what species that you have on hand is key to conservation. So where did all these new records come from? Some of our recent records came from the Connecticut Wild Bee Monitoring Program as a response, which I started in 2010 as a response to the growing concern over pollinator decline. We started with just 2 sites, one here and one at Lockwood Farm. And then we expanded to include other parts of the states and other habitats in 2011 and 2012. We included the preserve, the Stuart B. McKinney National Wildlife Refuge in Westbrook with the help of Chris Vagos, and a meadow in the highlands of Litchfield at the White Memorial Conservation Center with the help of Jamie Fisher. We also added the research station in Griswold in the eastern part of the state with the help of Rod Durgee, and the campus in Windsor with the help of Rose Hiskus and Diane Riddle, and who's here, and thank you, Rose, thank you, Rose. Morgan Lowry assisted with the main campus in Lockwood. In addition, I made an effort to survey in key habitats of greatest concern as defined by Connecticut Deep State Action Wildlife Plan. These habitats contain unique plant communities that host unique bee communities. One such habitat is the early successional habitat in wildlife management areas and power line rights of way. The early successional habitat has really important bee forage plants, which include staghorn sumac, sweet pepperbush, blueberry, sheep laurel, nannyberry, huckleberry, and winged sumac. There are certain bee species that are restricted to deep sand and/or coastal habitats because of their nesting preferences. If you haven't seen an inland sand plain, I suggest that you visit one because it's so astonishing to see what looks like a beach in the middle of the forest. Unfortunately, there are very few sand plains left in Connecticut, and it's estimated that 95% of our original sand plains are now gone. So other bees are restricted by the utilization of specific plants for pollen. Calcareous fens and riparian zones both harbor unique plants that are not found elsewhere. To find some of these specialized bee species, we targeted their host plants which occur in these habitats. In addition to habitats of greatest conservation need, we also surveyed vegetable farms, forest openings, and arboretums. Farmers are now incorporating pollinator plants on their farms, which improves the biodiversity of bees that visit their crops and can also help prevent soil erosion. Some farmers intentionally plant pollinator meadows adjacent to field crops or intersperse flowers between crop rows. For one project, we evaluated alternative floral resources on vegetable farms in Connecticut, and we looked on plants that the farms already had growing on their land, such as herbs, wildflowers, cut flowers, cover crops, and even weeds. We sampled bees on 10 farms over 2 growing seasons and found that these diversified farms supported 98 species of bees, which is roughly 26% of the bee species known for Connecticut. Many of the bees collected on non-vegetable host plants have been shown in other studies to be visitors of vegetable crops. We also looked in forests and forest openings. Surprisingly, a large percentage of the bee species in our region are forest associated. These bee species were likely the original dominant bee fauna in eastern North America prior to European colonization, when the region was extensively forested. Most are vernal species that have evolved to be active as adults before the tree canopy closes, when light levels are high and flowers are available. Important bee plants in forest openings include spring beauty, dogtooth violets, jewelweed, geranium, as well as red maple, mountain maple, black cherry, and willow. In our surveys, we primarily use 4 collection methods. We use trap nests for cavity nesting bees. We use pan traps, which are also known as bee bowls. They're painted with colors known to be attractive to the bees in our region, and they act like flower mimics. We also use aerial nets, and at one point we even converted a leaf blower into a giant insect vacuum. There are trade-offs for each collection method. Pan traps are cheap and can be deployed by novices at any time of day. They can be left out in inclement weather to run for longer periods of time, but they don't provide any information about floral associations. Trap nests capture only cavity nesting species. They also require no expertise and can give information about fecundity and pollen provisions. Aerial netting requires practice and patience, and observers differ in their ability to capture and extract bees from the net, which makes standardized collections amongst different people very difficult. Netting can be biased towards the larger, slower, and louder species if one lacks the expertise. Netting has the advantage of giving floral associations, however. Vacuuming has the same challenges as netting. However, a vacuum can be handy for collecting in brambles, such as raspberries, blackberries, and rose. After a few years, we opted not to use the vacuum in the photo anymore because it was cumbersome and loud and smelly from the gas, so we purchased a smaller dirt devil instead. Surveys which use all of the above methods in conjunction with each other capture a good representation of the bee community. Now for the results. What did we find? So as you can see, Connecticut has a very diverse bee fauna. We're up to 378 species, and that's not including the morphotypes whose taxonomy is still being worked on. The Halictidae, the sweat bee family, and the Andrenidae make up the bulk of species in Connecticut, with the Apidae and Megachilidae trailing behind. Melididae is a very small group of relatively uncommon bees. New Haven County had the greatest number of recorded species, followed by Hartford, New London, and Litchfield, Tolland, Fairfield, Middlesex, and lastly Windham. This is likely the result of relative collection effort in each county, reflecting the presence of Yale and our main laboratories and farm in New Haven. And at the other end of this scale is the lack of collectors in rural Windham County. There's 312 bee species in Connecticut are pollen generalists, which means that they'll collect pollen and nectar from many different types of plants. This is a bipartite diagram that I made for a study of the bee fauna of Grass Island, which is a preserve in Guilford. And it's a good visual to show the floral associations of bees. All of the bee species in this diagram are generalists. The diagram shows the interaction strength between bee species and the species of plants that they visited. The floral hosts are on the left and the bee species are on the right. The plants colored in aqua are specific to coastal areas. Bee species in the same genera are colored with this same hue. The length of the plant blocks represents total bee visitation observed on that plant species. The length of the bee species blocks on the right represents total bee visits observed to all flowers. And the width of the diagonal line connecting the bee species and plant species represents the number of bee-plant interactions observed. You can see that the plant species that had the most visitation and the greatest diversity of bees was beach rose, Rosa rugosa, and that Bombus impatiens, the common eastern bumblebee, had the most flower visits, particularly to beach rose. So 66 bee species in Connecticut are pollen specialists. These are bees that visit just one host plant species, one genus, or one family. This is important to know for conservation purposes because if we lose the habitat, then we lose the plants. If we lose the plants, we lose the bees. Pollen specialists may use other plants for sources of nectar for themselves, but they need the specific pollen to feed their larvae. The little bee here in my slide is the poster child for a bee species that everyone can help. They specialize on violet flowers. We've been taught that violets are lawn weeds and that we should kill them. Andrina violi, the violet miner, can only use violet pollen to feed its offspring. Violets are not a weed, so please leave the violets in your yard this spring. Another specialist bee in Connecticut is the hibiscus bee. It's rather uncommon and it's typically found near wetlands such as marshes and rivers. It was first detected in Connecticut in 1996 by the West River in New Haven and has been captured at the Pond Lily Nature Preserve in New Haven, at Hammonasset Beach in Madison, and even on the grounds here at the New Haven campus. We also found it nesting in a couple of baseball fields, one in New Haven and the other in East Hartford. This species is a ground nester and prefers compacted soils. They have a unique behavior of landing on water, which they collect for softening soil, for their nest, and for cell construction. They usually build a turret, but it had rained on the nest in the center photo, so you can't see it. It's leveled out. And then we have two very specialist bees in Connecticut. The first is a tiny mining bee. It's a specialist of chestnut pollen, meaning that it can only use the pollen from castanea flowers to feed its offspring. Since American chestnuts disappeared, there were no recent records of this bee species in the Northeast until 2019 in Connecticut. Andrina rennie was rediscovered in 2019 by Morgan Lowry in the chestnut orchards at Lockwood Farm in Hamden during a targeted search for the bee. So that discovery sparked a search for this bee species in other states and led to a more formal survey, which was conducted last year. We surveyed 27 sites in Connecticut, Massachusetts, Rhode Island, and New Hampshire. We looked in hybrid orchards, in forests, on individual trees, and there was only 4 sites where we detected this species from all those surveys. And one was Lockwood Farm. The other was Roger Torrey Peterson Estuary Center in Old Lyme, and 2 sites in Massachusetts. And I wanted to say a huge thank you to Dr. Agnes Dakis for all of the work that she's done to help the American chestnut tree and for maintaining the chestnut orchards, which has created a sanctuary for Andrina rennie at Lockwood Farm in Hamden. The other very specialist bee is a tiny mining bee, and it's a specialist on Parnassia pollen. And this was the first time that this rarely collected species was found in our state. I had heard from my bee friends in Massachusetts that they recently found this species in the Berkshires. So I made an effort with Claire Rutledge on one weekend to try to find it in Connecticut, which we did. The plant that this bee needs only grows in calcareous fens that are geologically restricted in Connecticut and whose habitat is threatened by Phragmites, nitrification, and development. This bee species is highly specialized on a plant that grows in an imperiled habitat, and its status will soon be listed as a regional species of greatest conservation need by the Northeast Fish and Wildlife Diversity Technical Committee. Certain bee species prefer coastal dunes and beaches, and they may also be in peril. As sea levels rise, storm surges and increasing storm intensity due to global warming are now threats to where these species live. There's 3 bee species in Connecticut. They're habitat specialists of coastal areas and dunes: Hylaea schwartzii, Lasioglossum marinum, and Lasioglossum georgic-wardi. There's also specialists of just sand and gravel and of interior dunes. The southeastern blueberry bee is uncommon in Connecticut, but shows limited persistence in the centrally located sand plains in the Connecticut River Valley. Its rarity now may be due to the loss of deep sand habitat. There are also other sand species that are not so particular about the type of sand that they nest in, and these are common throughout the state where sand can be found. There are 4 bee species that are currently listed in Connecticut. These status assessments were made in 2015. The rusty patch bumblebee was the first bee to become protected under the Federal Endangered Species Act in 2017. It was last collected in Connecticut in 1997 by Chris Meyer. Bombus ashtoni is a social parasite of the rusty patch, so it makes sense that its decline parallels that of its host. It was last detected in Connecticut in 1992, and I believe that collector was Mike Thomas. The yellow-banded bumblebee was last detected in 2021, and as you'll see in the next slide, there is hope for that species recovery in the Northeast and in Connecticut. The Macropus cuckoo is a very rare bee. It was last detected in Connecticut in 2006 by Dave Wagner in his surveys of powerline rights-of-way. Right now, I'm working with NatureServe and Connecticut Deep to update this list for the 2025 Connecticut Wildlife Action Plan. The Bombus terricola may be recovering in the northwest corner of Connecticut. Prior to 2018, it was last detected in Connecticut in 2009. Recent surveys have found 18 individuals since 2018 in Litchfield counties. So other New England states also see similar trends, and they're finding increasing numbers. There's also bad news. So among other bee species that will need our attention, these 3 are top priority in Connecticut right now. So there's a petition to list Bombus pennsylvanicus as a federal endangered species, much like the rusty patch, which was listed in 2017. So Bombus pennsylvanicus, which is also called the American bumblebee, was last detected in Connecticut in 2006. It prefers to nest in grassland and open farmland. Bombus has only one record since 2010. This species is very mysteriously absent from other New England states as well. And what makes this strange is that it's a social parasite of two bees that are very, very dirt common. So we don't know what's happening with that right now, but we're trying to figure that out. And Bombus ternarius is a northern species whose range may be contracting in Connecticut and moving northward as the climate changes. To address those species and other species of concern, I'm going to be working with NatureServe and Connecticut Dept to give conservation ranks to our Connecticut bees using NatureServe's systematic, standardized, and quantitative methods that are used throughout the US and Canada. Species will be given a rank of either S1 to S5, with S1 being the most imperiled and S5 being the most stable. So when making conservation assessments, there's a few things that have to be considered. In order to evaluate whether species are in a decline, you have to have a knowledge of the habitat requirements. You need to know the past distribution to see if you're on the edge of a range or if the range might be contracting. You need past occurrence data, or in other words, look at the commonness or rarity of a species. You must also collaborate with other nearby states and look at things from a regional perspective, and look at the potential environmental threats, such as imperiled habitat or climate change. The other important point is that just because you can't find a species does not mean that it's not there. It's more clear-cut to validate that a species persists than to prove that it does not. For instance, 142 species that were reported in all of the early checklists have persisted until at least 2011, when looking at records from 2011 to 2021. On the flip side, about 92 species have specimen records spanning 1864 to 2011, with no records past 2011. So does that mean that they're not here anymore? Not necessarily. It's a very tricky thing to figure out, and I'll be doing that using NatureServe's rank calculator. I suspect a good majority of these records are data deficient, which is when you have to look at other factors, like are they a generalist or a specialist? Is the host plant that they prefer in danger of extinction? Is the habitat that they prefer being destroyed? Oh, wait a minute, gotta go back. And the last point that I want to make is that it's so very important to preserve specimens even after they're published or put into digital databases. When making conservation assessments about species, you must be able to re-examine specimens to verify the ID. Luckily for us, a lot of the specimens that Viereck and Britton used from 1904 and 1905 in their initial checklists are still in our collection. So we were able to re-examine them to verify their identifications, as many of the names have changed since people have revised the taxonomy. Museum collections are a vital part of the historical record and must be valued and preserved. So in summary, in the course of creating this checklist, we've collected over 6,000 specimens in 99 towns across Connecticut. We've documented 378 species for the state. We've identified key habitats that are important for bee conservation. We have documented what their phenology is in the state with their distribution, which will be important as the climate changes. So finally, this research provides a foundation for future research into bee conservation and ecology. And my list of people to thank is so enormous because I have been working with everybody for 10 years or more. So I just want to say thank you to everybody who has helped, everybody who's participated and has taken the time to collect the bees and go out and check the traps. And yeah, it's just been a really amazing trip for me. It's just been great. And so just want to thank you all from the bottom of my heart. Thank you. So I'm open to questions. Yes, Chris?
Your sampling techniques are all sort of ground sampling techniques. What do you think is up in the canopy that we missed?
That's what we're trying this year. We actually just put some of our trap nests up in the canopy at Litchfield. And we're part of a larger experiment that's going on with people in Georgia, Florida, New Jersey. I think there's 7 different states cooperating, and we're putting trap nests into canopies. And so this is the first year that we're doing it, which is just basically to test various types of trapping. We made some branch traps, and we also use traps made from lumber from Home Depot. And then after that, next year, the idea is to expand even further and possibly get people from other countries involved as well. And we want to see if the vertical gradient, if the bees differ in the canopy from the mid canopy to the ground level. And
What about the non-inventors?
we could get into that. There are people who were doing pan traps up in the canopy as well.
Just an aerial malaise or canopy.
Mm-hmm.
Canopy probably would, although with the winds we've been having, very challenging.
Yeah, yeah. That is true. Huh? Yeah. Yeah.
Are there any of the bees that are truly crepuscular, just active at dusk and dawn?
The dawn bees I know. Oh, I'm sorry. So the question was, are there any crepuscular bees in this state? Meaning, are there bees that come out only at dusk or at dawn? And Pepinapis prunosa, the squash bee, is a very early riser. When we were doing our studies, the field assistants were going out at like 6 in the morning to Lockwood Farm. And for nighttime bees, there's a species of lazy glossum that feeds on evening primrose, and they come out at night. Yeah, those are the only 2 that I know of. Yep.
Thank you very much for the moment seminar. Do you know if there is any case of any of those paralysis viruses in the state of Connecticut?
So the question was, are there any viruses and diseases in the state of Connecticut in our bees?
If there is any report, because I know paralysis viruses are everywhere, but they have been reported yet.
So Tracy, there are these honeybee paralysis viruses that have been studied a lot as sort of the complex of honeybee viruses. So I think, and we've done surveys of viruses in honeybees and found some of those viruses, I think.
Correct.
Yeah.
The wing is the most common.
Yeah. So I don't, as far as I know, there's not anything known about those paralysis viruses in the wild bees. There are other viruses that we know that are common between honeybees and like bumblebees or other kinds of bees, but I don't think the paralysis viruses are among them.
Neil?
Follow-up question.
Well, they visit the same flowers. So like honeybees could be on the same flowers as our native bees. And then the viruses could be deposited on the flowers, and then native bees could go visit the flowers and then pick it up from there.
Yeah. people
And from
have
their
studied
pollen.
That possible route of transmission of viruses spreading from different, among different species of bees on the flowers. I looked at like the structure of flowers and how that might affect it.
Mm-hmm.
So they really don't get baseline. Secondary. Eating, yeah, flower.
I'm not sure I understand the question, and you're kind of muffled.
Bees, solitary bees and honeybees.
Yeah.
Just don't get- yeah, flower.
They do. If you, we have lots of videos of honeybees and bumblebees interacting in squash flowers. Yeah, and they'll hit each other and bump into each other, and sometimes one bee leaves, sometimes another bee leaves. Yes, we were trying to figure out if one species was more dominant, like a big bumblebee would come and kick out a honeybee, or vice versa. And yeah.
Thank you for the interesting talk. I was wondering, apart from the impact of climate change on habitat, rock soil, whether there are any additional evidence that climate change directly or indirectly causing or differential, having a differential impact on different bee species in Connecticut. In other words, whether changes in temperature is causing some bee species to be more active and other bee species suffer as a result of direct impact of climate change.
So the question in a nutshell was, how might climate change be affecting the bees in Connecticut? And what we're seeing, we're starting to see some more bees that are more of a southern species. We're picking them here, up here in the state now. So the species ranges, from the south are expanding upwards into our state, and then some of our more northern bees that are only up in Litchfield County are starting to move even more farther north. And especially bumblebees, because they're, I think, more sensitive to temperature than others. So, are there any other questions? Yes?
I'm just curious, when you talk characterization, are you relying solely on homology, or do you guys do any other molecular studies part of the characterization?
So the question was, when I identify the bees, do I just use the morphology or DNA testing? And I have sent some species up to Canada. There was one really interesting Hylaeus bee that I found on Grass Island, and a fellow researcher, John Asher, also found the same thing on Gardner's Island in New York, and it looks like another bee, Hylaeus schwartzii, but it's a little bit different, and it keys out to be a species that's in wetlands, but not necessarily like marsh kind of wetlands. So we are still trying to figure out what that species complex might be, but for the most part, it's morphology. And I have a lot of people ask me, why do you have to kill the bees if you're trying to save them? But a lot of the features are just, you can't see them with the naked eye. You have to look at the punctures, you have to count the hairs, you have to see the hairs under different lights, look at the mandibles, so. Any other questions? I saw you had, Chris, you had your hand up earlier, but
I'll ask this.
okay, that's okay.
Going back to the boom and bust cycle of some insect species, I suspect there's very little known about bees that might do that. I mean, there are moth species, for example, that are only common once every 50 years. We can see where we might misinterpret that species is really rare or gone.
Right.
Has it been documented, even if it's only like a 10-year gap between bees? Do any bees have a boom and bust cycle that's been documented? As well as it can be, of course.
I don't know if it's been documented. I know that we found that black and gold bumblebee last year that hasn't been seen in Connecticut since the early 1900s. And then all of a sudden in Milford, it showed up and somebody took a picture of it and they posted it to iNaturalist and was like, wow, like what the heck is this thing? And then somebody else saw the same species. I don't know if it was the same bee or not, but they saw the same species in 2 other counties. So that could be a boom or bust phenomenon. I'm not sure.
Maybe
But the
it's linked to a host.
they were all found on Monarda, which is interesting, in all 3 counties last year.
Just a plant people are planting all over the place now that didn't used to be anywhere near as common. Basically, I think absent in the southern state. Now it's in garden.
Yep. Do you have any questions from the chat?
No, nobody has submitted any questions for me.
Okay. All right, then. I guess that's it.
All right, thanks, Tracy.
All right, thank you.